Shri Gurubhyo Namaha. Welcome to our online teaching academy Ekalaiva, and I welcome you all for the biology class. This is class 12 biology. We are in the second topic, biotechnology. In first topic already we completed the principles, and here we are going to talk about the application. So biotech and its application. Before stepping into its application, let us see the critical research areas, the three critical research areas. Providing the best catalyst. Okay, actually you would have read about the enzymes, the different types of enzymes, how they are preparing, and then rDNA technology. So the first thing, providing best catalyst, okay, and then organism, usually microbe we are using in this technology. Yeah. And then creating optimal conditions for the growth and the development of the vaccine or whatever it is. Then downstreaming process by using bioreactor, etc. So these three critical research area, important 2-mark question. Now we'll see about the different types, okay, in application, in what are the different fields they are applying this biotech. So the 4 fields you can see here, one in agriculture, healthcare, environment and industry. These 2 fields are at the peak now, and now in this pandemic, okay, we are talking about vaccine, right? So we are going to learn more about this in this lesson. So another one, environment, and then industries like preparing enzymes, etc. So the code, each one have different code. Like red biotechnology is medical, white biotechnology industrial, then green biotechnology agriculture, blue marine, the different field. So this field we are going to see in detail first, and then we'll move to the green field. So biotech in medicine, I'll show you the image. Here one thing is highlighted, and this is very important. You would have heard this one in 11th standard also. This particular gland acts as the heterocrine gland. It produces both enzymes as well as the hormone. This hormone, when it is not functioning well, when it is not coming out in a precise quantity, there are some defects. So what are they? That we are going to see. And this is a glucometer to check whether the hormone comes in a precise amount. So before that, let me see this one. The gland, how they are going to synthesize and why they want to synthesize the different types of hormone. What we read in 11th standard, a small glimpse we'll see. Pancreas has different types of cells, alpha, beta, delta, gamma, like that. So in the 2 major cells, they are going to produce very important hormone like glucagon and insulin. This glucagon role, it stimulates the glycogen, and then it raises the blood sugar level. So the blood sugar level will be more. Then what happens when there is a reaction like this? It triggers the gland and it asks to produce insulin. Now this insulin will lower the blood sugar level. So it is an antagonistic mechanism, feedback mechanism. One is going to high, another one going to be low. It has to be in a regulated way. Sometimes what happens, when the blood sugar level is more and it's going to trigger and there is no production of insulin, in that case it leads into diabetes mellitus. So for that person we need artificial thing. So how to inject it, from where we want to prepare, all these questions was keep blinking. And then later the industry, Eli Lilly company, they came out with a very good solution called Humulin. Right here. Humulin, okay, an artificial insulin, okay, which they can inject. So this is the structure of insulin. It is another 2-mark question, the structure with explanation, 3-mark also. If you see the structure, it made up of 3 chains. One is A, B, another one C. Okay, let me write like this. I am writing one chain in between. Actually, it is not A, B, C, it is A, C, B. It is made up of polypeptide chain, 2 chain with a linker chain. This you call it as linker. This chain is a linker chain. A, B chain, they are very important, and that chain, those two chains, they are linked with the disulfide bond. This is the structure. One is proinsulin, which is not active, okay, it's a precursor form, and then it has to be activated, it becomes insulin here, activated insulin. You can see the structure and draw the structure with the correct sequence A, C, B, like that, right? And the chain, A chain is made up of 21 amino acid, B chain made up of 30 amino acid. So here is the preparation technique. We'll see that once. Yeah. So I told you about A chain and B chain. Okay, how they are going to synthesize these two chains, and artificially they will be bond up, okay, with a peptide bond. So first they will take the gene from A and then from B. Okay, you can see here. First let us see according to the picture. Okay, this is the bacteria. In E. coli bacteria, they are going to synthesize. You can see the A chain and then the B chain. They will fuse with the plasmid. And then what happens? Separately they will synthesize the chain, and then later it will be incorporated together and it become active insulin. Okay, directly they are injecting into the body, so it won't be in a proinsulin form. Okay, there is no requirement of C chain, so directly it will be in A and B, and they'll bond up with the disulfide bond. So this technology already would have read in rDNA technology, the same process only here, rDNA technology. Okay, and you need not explain the rDNA technology here. Here totally we are focusing about how they are producing the insulin. Okay. So by this method, and what is humulin? Humulin is the first insulin which we are synthesized from the human. So we call it as humulin, right? Next, another important thing we are going to see, monoclonal antibody. The name, first let us understand the term monoclonal antibody. What is mono? Mono is single. Clonal, something clone what you say, identical. Something is going to be identical. So from one thing, okay, something going to be identical we are going to produce. So what it is? That is nothing but antibody. Okay, biotechnology, hybridoma technology. So how are we going to synthesize the antibody? Okay, I will tell you one example. For example, if you are preparing some vaccine, polio vaccine or some other vaccine, if you prepare, we will take that particular microbes, and that microbe will be in a suppressed form. Okay, and that microbe only injected in our body as vaccine. Now, any antigen enters our body will react against that. So, what it produces? It produces antibody. Okay, like fighting mechanism, it produces antibody. So, like that only the vaccines are prepared. Suppose in that vaccine, if any one microbe is going to be alive and that is going to enter your body, then it leads into this disorder. What you are taking, that disease you may get. Okay, to avoid that, we are using here another technology called monoclonal antibody technology or antibody technique, right? So, how they are going to do that, we are going to see. So, monoclonal antibody are a laboratory produced molecules. Okay, they substitute the antibody and they provide immunity. So, here is an example, okay, and how they are doing the technique. So, from the animal, okay, from the animal they will take the cell. So, which cell they are going to isolate? They are going to isolate the immune cell. The immune cell are lymphocytes. Okay, and this lymphocytes will be activated. It will be triggered by introducing that particular antigen, and it start producing the antibody. And this cell plus there is another cell from the bone marrow. We call it as myeloma cell or tumor cell. Okay, and this cell, what is the unique feature about this cell? They can propagate very rapidly. So they will take this one myeloma cell plus our lymphocyte. Okay, together we call it as one cell. It is a hybridoma cell. Okay, the name you can see here, hybridoma cell. Okay, in this cell, now what happened? It will produce the antibody according to the antigen what we have introduced there. And the ability of producing or propagating okay, in a rapid way by myeloma cell will also be induced here. So, both the character will be there. We will get many number of cells with the desired antibody, and this technique you call it as monoclonal antibody. So, what are the topics we have completed? The first topic is about insulin, the structure of the insulin and how they are preparing the two different chain, A chain and B chain, and another one monoclonal antibody. Next class we will see about this syndrome. Okay, the baby is inside something, right? Bubble boy syndrome we are going to see, right? To receive our online lesson, please press subscribe button and you will receive our latest updates. Thank you.